Angelica Faccoretti. Angelica graduated in biological sciences at the University of Pavia and holds a PhD in cell biology. In 2006, she obtained the European Master degree in radiation biology at the University College of London and subsequently the title of specialization in clinical pathology at the University of Pavia. Since 2010, she is responsible for the radiobiological experimental activities at the National Center for Oncological Hadron Therapy, CNAO of Pavia. She is adjunct professor of applied biology at the University of Milan for the School of Specialization in Medical Physics and of Biology, Anatomy, and Physiology for the master's degree in Physical Sciences at the University of Pavia. Angelica is also very much involved in the management of the HITR Plus project, and it is a pleasure to work with her. And it's also a pleasure, Angelica, to give you the floor for your lecture. Thank you for taking this slot.
Thank you. Thank you very much, Nicholas, for your kind introduction, and also for giving me the opportunity to be among the organizers of this course. I just ask you to be so kind to let me know when I have 5 minutes left, just to be sure not to go too much over my time, please.
Yes, of course.
Okay, so let's talk about radiation biology for hadron therapy. Thanks to Eleanor Blackley that yesterday she gave a very, very interesting introduction on radiation biology, on the historical aspects of radiation biology for heavy particles and high energy radiation. I have this very great advantage today to introduce you some of the aspects of radiation biology for hadron therapy. This is an overview of my talk. Actually, I'm going to divide this lecture in 2 parts, trying to keep it as much interesting as possible and also to be a little bit provoking for some aspects. So I will start with what I called the classic radiation biology for hadron therapy. So I'm going to give you an overview of the classic terms of radiation biology for hadron therapy. So LAT, RB, oxygen effect, and all that aspects that we have already touched yesterday, but I will go a little bit more in detail. And then I will leave the last part of my talk to the what I called the rock radiation biology. As I said, is a sort of provocative term, just to be on the other side of the classic radiation biology, which is, let's say, the most historical part. And I'm going so with the rock radiation biology to illustrate you some aspects that are rising in the radiation biology field, and that I hope that will be the main topics of the next future. I will start. Before starting, I need to make a disclaimer, because I know that probably each one of you has a different expectation from this presentation, due to the fact that radiation biology has really many different meanings, because it is a very huge field with very different topics. And here I listed just some of them, and surely I will not be able to touch all these aspects, but I will try at least to give you some indication and some good point for you starting to be interested at least in radiation biology, or at least to increase your curiosity on this topic. So please just apologize myself whether I will not be able to cover all this aspect. So let's start, as I told you, from something that is really, really classic, because we cannot start to talk about radiobiology for high LAT radiation without talking about LAT. That is the linear energy transfer. You have already some notion yesterday. The LAT is the amount of energy that a particle is able to transfer during its traversal on the material per unit distance. And I'm not a physicist, as you heard, I'm a biologist, so it's quite clear even to me the difference between the ionization density profiles of a proton versus a carbon ion. And let's say that everything can start from here. As yesterday Eleanor said, not everything is explained by this, but can start from this at all, definitely. So LAT, from a biological point of view, from a biologist point of view, describe the action of a radiation into the matter. But what is the matter? If we talk with the physicists, of course, the classic definition of LAT is the matter is something that is water equivalent. But if we move to a more biological or clinical point of view, the matter is a patient. So of course, if we have a patient that is under a treatment, the beam will traverse the patient and will have effects on very different levels of his bodies. So starting from the organ system to organs, tissues, cells, and molecules. The classic radiation biology has focused a lot of its attention on the molecule side, on the molecule level, because obviously everything started from the interaction of the radiation on the molecule. And that is the reason why for several decades, when we talk about radiation, the classic, let's say the conventional radiation biology and the radiation biology for hadron therapy, we have focused our attention on the DNA damage. Why? Just simply because the DNA is the most important molecule inside the cell, because it cannot be replaced, whereas on the other side, all the organelles actually can be replaced by itself. And on the other side, the DNA is the most critical molecule for the survival of the cells, but also for the destiny of the future cells of the progeny. So that's why we have started, and a lot of biological studies in radiation biology are performed at the DNA level. But just let's go to have a see on what is happening when highlight radiation traverses a cell that is mainly made by water, of course. So the highlight radiation ionizes the water that is inside the cell, and it will produce hydrogen atoms and hydroxy radicals in a very, very short tracks, because we are talking about highlight radiation. So in this case, there is a high probability that two events will occur in the same cell. And so for that reason, there will be then the formation to peroxide inside the cells, and this peroxide will induce a damage to the cell or to the molecules, in this case to the DNA. When we consider low LAT radiation, we have a very high probability that two events will not occur in the same cells, because the ionization events are far from each other. So actually, the radicals that will be formed will be then reunited to reform water. So no damage will happen in this case. And if we move to the DNA level and we see what these events will occur at the DNA level, we can see this very nice diagram that you have already seen yesterday, where sparsely ionizing radiation or densely ionizing radiation are overlapped on the DNA molecule. And it's quite clear how the difference is due to the fact that we have many ionization events on the same DNA molecule. So what happens at the nuclear level? We can see here a nucleus of a cell with different green spots. They represent the places where the DNA damage is going to be repaired. So actually, with low LAT radiation, we have isolated damages that can be quite easily repaired by our cells pathways. When we move to consider highlight radiation, so for example, carbon ions, we have a totally different view. We have a very big damages. We have definitely a big holes inside the molecule of the DNA. And these are called cluster DNA damages that are very difficult to be repaired by the cells. And for that reason, we will see delayed DNA repair or misrepair. And so the cell will have definitely many problems to go ahead. So another classic parameter that is considered when we talk about radiobiology for different radiation than photons is the RB, that is the relative biological effectiveness. Actually, it is a parameter that is able to give us an idea of the power of radiation to induce a damage in the cells compared to photons. So is a ratio between a dose of photons versus dose of another radiation to induce the same ISO effect. So it's a very useful parameter to give an idea, as I told you, of the power of radiation. But please, I want to give you some warnings on the fact that it can give you just an idea, but the power of radiation needs to be definitely investigated more into the detail. So for carbon ions, there are really many different RB values that have been collected during the history of radiation biology. And actually we have ranging from 2 to 5, but we can also find even higher values for RB. On the other side, we have an empirical fixed RB for protons that is 1.1, and is the value that is used in clinic. Actually, as I told you, is an empirical is fixed, but there is a lot of problems, let's say, behind this value, because experimental speaking, different values has been collected in different conditions. So actually is a matter of study, and I hope also of debating and changing in the few years. So as I told you, RB is a very nice and useful parameter to start to evaluate the effects of radiation. But please be careful on the fact that this parameter is affected by very many different experimental conditions. So it is affected by the radiation quality, of course, but by energy of radiation, by the dose rate, by the level of the dose, the number of fractions, the biological system that is considered, so starting from one cell to the other one, and also to the endpoint, because so far we have talked about RB considering the classic RB on the cell survival, on chronogenic cell survival, but actually we can calculate RBE for all the biological endpoints that can be investigated. So just please, when you refer to RB, be careful to have well-defined experimental condition and to have clear what is going behind. And another warning signal that I want to give you is that when we calculate RB for chronogenic survival, we consider with the same level, with the same importance, a colony like this one and a colony like this one. So this one, just to highlight you the fact that there should be something more behind the formation of a colony, because there should be different biological mechanism for which a cell was able to produce only this such small colony instead of this one. So again, we have to go deeper into the details. So this is a classic graph representing the value of the different survival curves of low LT radiation and high LT radiation, and considering the different length. So for example, For photons, we have the classical cell survival curves, Schalder cell survival curve. And when we move to highlight radiation, we have a definitely linear steep curve, meaning that higher is the let, higher is the power of the radiation to induce clonogenic cell killing. But so from this graph, it seems that higher is the let, higher is the RB. But is it valid always? No, you already know that yesterday we have seen that is not valid always, because the relationship between LAT and RB change accordingly to this graph. So for radiation with the low LAT, lower than 10 keV on micron, actually the RB slowly increase according to the LAT. But then we have a very high increase, rapid increase of the RB for radiation with the LAT between 10 and 100. And then we reach the peak at 100 keV. And then it again fall down for radiation that are high, with higher LAT of 100. This is due to the fact that the distance of the ionization events that we have at 100 keV per micron is equal to the diameter of the DNA. So it's about 2 nanometers. So this is the best condition to have cluster and double strand breaks on the DNA. When we move to higher LAT values, actually we waste energy because the events will coincide one to each other. So actually we will not so good in inducing the damage to the DNA and referring to the energy that is deposited. Again, here we are talking about DNA, and we are assuming that in this condition, the double strand breaks are the basis of most biological effects. So actually it is true, because we said that DNA is the most important molecule, but please consider that this graph is surely valid for cell killing, mutagenesis, or oncogenic transformation. But actually, we don't know whether this is valid also for other biological events that will deal with other organelles or other molecules into the cell. So just please into consideration this, and I hope that we will in the future have updates on this direction. Another classic point of hadron therapy, of radiation biology for hadron therapy, is how highly radiation differ in respect to the cell cycle phases. We know that with photons, we have different responses from the cells according to their status in the cell cycle. So we have a high radio resistance for cells that are in the final stage of the S phase. On the other side, they are more radiosensitive in the mitosis phase. This is due probably to the chromatin condensation that makes the DNA repair difficult during the mitosis. When we move with the highlight radiation, this dependence is not so evident, probably due to the fact that the big damages that are induced with this radiation are not dependent by the chromatin condensation or by anyhow the conformation of the DNA inside the nucleus. Other point, oxygen effect. We have heard yesterday about that. Here is a clear histological imaging of a section of a tumor, where it is possible to see the vessels and the hypoxic region far from the vessels, and then the necrosis region. So this means that inside the tumor, we have many cells that are in a hypoxic microenvironment. And actually, these cells, we know that are more radio-resistant to conventional radiotherapy. So this is explained in, again, with the cell survival curves in anoxic and aerobic environment. So you can see the difference in the curves. And this difference can be quantified by a parameter that is the oxygen enhancement ratio. That is the ratio of hypoxic to aerobic doses that are needed to achieve again the same biological effect. So as you can see, even the oxygen enhancement ratio depends by the dose level. But why do we have such a difference in response after radiation in hypoxic and aerobic condition? This is mainly due to the fact that under aerobic condition, the radiation-induced radicals are able to react with the oxygen that is present into the cells and in the microenvironment, and this will induce then damage to the DNA. In the case of hypoxic condition, so in the case of lack of oxygen, the radicals will be then reduced and will not induce any damage to the DNA. So this is the basis of the so-called oxygen fixation hypothesis. And when we move to see the effects of oxygen concentration after highlight radiation, so for example, carbon ion radiation, we see that the differences in the response is not so high. So the oxygen enhancement ratio, for example, in this case, dropped from 3 to 1.5. Why do we have such a difference in the response? Actually, we have to take a look on the different modalities of action of the radiation at the level of the molecules, and in particular at the level of the DNA. So with low HITR radiation, we have as dominant the indirect action. So we have the formation of free radicals, then they will react with the DNA. And as I said, without the presence of the oxygen, there will be not the fixation of the damage on the DNA. So we will not have double strand breaks. When we talk about highlight radiation, actually, the main important action is the direct one. So we don't have second messages to induce the damage to the DNA. And so the oxygen is not needed to fix the damage to the DNA. So that is the reason why we don't have such a big oxygen effect with highlight radiation. So finalizing the classic, the DNA-centered radiobiology, actually what can happen to irradiated cells that are facing to big damages to the DNA. The cells can have no effect when actually the cell is able to repair the damage, or there may be division delay, so the cell will be stopped in dividing through cell division, or we can face to apoptosis of the cell, so the cell will physiologically die because it's not able to repair or to restore the damage anymore. We can have the so-called reproductive failure, so the cell is not anymore able to go further into mitosis. We can have genomic instability, which is actually something that is more related to a delayed form of reproductive failure. Or, of course, we can have mutation into the cell that will survive and will keep the mutation into the DNA. And we can have oncologic transformation that in the case of clinic, of course, we take care more about oncological transformation. And then the bystander effects and the adaptive response. I'm not going to touch this into the detail, but actually what we know now is that an irradiated cell is able to send signals to the neighboring cells that are not irradiated and to induce a genetic damage in them. Not only genetic damage, let me say, but in this case that we are considering DNA damage, DNA-centered damages is the main important. And then we can also face to adaptive response. So a cell that has been irradiated actually with very low doses is then able to react in a more radio-resistant way to subsequent exposure to radiation. So far, we have said that DNA is important, and it is definitely important, and thanks God that we have collected a lot of data on the effects of different radiation at the DNA level, and they are, this information are the basis for all the clinical aspects of our beams. But actually, we have now, it is now time to take into consideration that ionizing radiation can deliver very important effects only also at other different organelles or molecules inside the cell. So, for example, to the ribosomes, to the endoplasmatic reticulum, or for example, the signal transduction pathways, and also to the mitochondria. So that's why I'm trying to be a little bit, let's say, provocative, but just to highlight the fact that it's really time also to consider other effects than the DNA damage-centered one. And so that's come my second part of the presentation of on rock radiobiology. So let's start, for example, to watch at hypoxia-induced radioresistance from a different point of view. The so-called biological oxygen effect. We have seen the chemical oxygen effect, and now let's see the fact that actually cells that are in hypoxic environment have a totally different metabolism respect to the normoxic cells. So here are listed some of the different pathways that are activated and that are present in hypoxic cells that may lead to an increase in radioresistance. So just taking one on all these, we have an increase transcription that is mediated by hypoxia-inducible factor 1, which is a very well-studied factor that is high expressed in hypoxic environment, and is one of the most important responsible of the so-called biological oxygen effect. So please take into consideration that beside the oxygen effect, we have all these and probably many other aspects that have to be considered. And here is a very nice study that has been performed in vivo on experimental model using a tumor, lung tumor, that were, let's say, normal lung tumor and the tumor with the lockdown of the hypoxic inducible factor. So actually, we can see the different response to X-rays of the two models, in meaning that in the case of the effect of hypoxic condition really affect the response to X-rays. On the other side, there is no effect on the lockdown, on the silencing of the HIF1 when we consider carbon ion. So this is a good point towards the fact that high-lit radiation, or at least carbon ion radiation, is able to overcome the biological, one of the aspects of biological radioresistance due to hypoxic regions. So this is a cartoon where they try to explain the pathway, the different pathways that are activated after carbon ion radiation and after photon irradiation. So actually, with the carbon ion radiation cells will not activate the mTOR pathway that is related to HIF. So there is a totally different response with the inhibition of tumor cells compared to photons where we have to pass through this pathway. So this is one point in favor of the fact that we have something more, as I told you, than DNA damage. Then another hot point when we consider radioresistance of tumors is the p53 mutation, or in any case, the resistance that many tumor cells has to go through apoptosis induced by exposure to radiation. We know that p53 is the guardian of our genome, and that is activated when we have damages to the DNA, mainly in our case, double-strand breaks. And then after the activation of this gene, we have a cascade of other messages that then will give the response, mainly growth outburst, apoptosis, DNA repair, or senescence. So all responses that will protect the cells after the exposure to the radiation. Unfortunately, we know that the majority of the tumors display mutations or lack in P53 already in the early stages of the development. So this started to be a very big problem in radioresistant, in intrinsic radioresistant of the tumors. And what has been investigated and what is going to be investigated in the future is the fact that highlight radiation is able to induce apoptosis in the tumor-radiated cells, activating a different pathway with respect to the P53. So actually, it has been, for example, described in this paper that highlight radiation will activate a caspase downstream, not through caspase 8, but through caspase 9 that pass through mitochondria. So we will in this case, the P53 pathway will be bypassed, and so we will definitely have successful in induce apoptosis in the irradiated cell. Another hot point that is investigated in these years is the different response regarding immigration and invasion of tumor cells after radiation. We have already collected many data saying that at least the low doses ranging to up to 4 grays of photons are able to induce migration and invasiveness of the survival tumor cells. And actually, this might be a problem in clinical because the cells that are in the, say, near the tumor cells near the tumor that receive very low doses may be able to increase their malignancy and their ability to produce recurrences. Actually, several works have studied to investigate also the role of carbon ion highlight radiation on the modulation of migration and invasion. Unfortunately, the data that have been collected so far are not enough to write a clear conclusion, but the tendency, let's say, is that carbon ions will not induce an increase in migration and invasion of irradiated cells. And some researchers has tried to find an explanation on this, in modulating and modeling the distribution of reactive oxygen species after radiation with photon and after radiation with carbon ion inside a cell volume. Here you can see the totally different patterns of distribution that is definitely diffuse in the case of photons, where for carbon ions are very well localized and dense along the ion path. So actually, what has been hypothesized is that this different distribution may preserve the plasma membrane and the intracellular structures of the cells that are outside the tracks. So in this case, this will not affect and are not enough to activate signaling pathways that may be involved in migration and invasion. So actually, the damage is not so widespread on all the cell structure, and so the pathways that are activated are totally different in these two situations. And I think that this is a very good point where to start if we want to consider the different cellular effect on after irradiation with carbon and photons, this different spatial distribution of the reactive oxygen species. Other point of the response of a tumor after carbon ion or photon irradiation is definitely linked to angiogenesis. This is a research paper that has been published to study the effect of photons versus carbon ions on a tumor that has been irradiated, and then where they try to investigate the architecture of the new vessels and or the old vessels after radiation. And here it's a very interesting graph where you can see that the number of microvessels after the radiation with photons is definitely increased compared to control and to carbon ion radiation. So, and it's even quite evident from these images. So again, another point that has to be investigated deeper into detail is definitely the angiogenic versus anti-angiogenic properties of the different radiations, because we know that microenvironment is definitely as important as the tumor cells when we consider the fate of a tumor after radiation. Then here are some highlights on the fact that we have also to consider the fact that after irradiation with photons, we know that tumor cells show a certain grade of activation. So, for example, we have seen an increase of melanin production on malignant melanocytes that have been irradiated with photons. But for example, we haven't seen the production of melanin after carbon ions. So again, stressing the fact that we are talking about different mechanisms that are activated and also different responses that the cells will activate to defense after the radiation. And on the same line, also the fact that carbon ion irradiation does not induce, for example, the dendrites formation and elongation on the same cells of malignant melanocytes, whereas here it's clearly evident morphologically from these images. And then again, if we want to talk about cellular aspects, we know that the different type of radiation can induce can activate different patterns of tumor cells irradiation. So another crucial point that has to be investigated and that is going to be investigated is which are the advantages of one radiation versus the other one in respect to this different pattern. Then let's see the topic of cancer stem cells. Cancer stem cells are cells that are present into tumor mass. They are very radioresistant and very chemioresistant. And if they survive, they are able to restore the tumor mass. So actually, the real target of tumor treatment should be the cancer stem cells, because otherwise, if we will be able to destroy the cancer stem cells, the tumor mass will not be able anymore to survive and to replenish the cell population. So a lot of studies have been conducted and are already in course to try to find a treatment that is able to overcome the radioresistance and the chemoresistance of these cells. And actually, from the data that we have on our hands on carbon ion or charged particle radiotherapy, we can say that probably this type of radiation is more effective in eradicate the cancer stem cells, because actually this, for example, these cells live in the so-called in hypoxic and microenvironment. So we are already seeing that carbon ions are more efficient in overcoming this radioresistance. They have an increased defense to the reactive oxygen species, and they also have activated many, let's say, defense mechanisms to avoid apoptosis. So actually, from what we have said so far, and so what we have collected with the research, we can say that probably heavy ion or high latent radiation may be overcome this radio resistance, but again, we need to go definitely deeper into this topic to see whether we can exploit the feature of high latent radiation to overcome this aspect. And then let's go to an even higher level, because we can talk about the effects that radiation can have on the immune system of the patient. We know that immune system play an important role in controlling the tumor growth, and actually a treatment should to treat a tumor should preserve at least the immunocompetency of the patient. So we know
5 minutes left, Angelica.
Thank you very much, Nicholas. So we know that the conventional radiotherapy, from one side, activates some factors that are immunosuppressive, but from the other side, we have also positive effects of radiotherapy on the immune response. But unfortunately, these positive effects are not sufficient to increase or to shift the balance of the response of the immune system of the patient in favor of cure the cancer. So one of the main aspects that is going to be covered in the next years is definitely the investigation to test whether the biology of high latent radiation may help to shift this balance, so may help the body to overcome the immunosuppressive factors that are released by the tumor and by the normal radiotherapy. So far, we have collected different data on this direction. So, for example, we know that carbon ion radiation is able to increase high mobility group OX1, which is a molecules that indicate the immunostimulant properties of carbon ions. On the other side, we have also activation of immunostimulating cytokines, which are definitely a good point in favor of a total body response against the tumor. And also, as I told you, high latent radiation may induce different cell death pathways compared to photons, and most likely these cell death pathways are more immunogenic than the ones activated by photons. So, and not less important, also the fact that thanks to the physical properties of high latent radiation, we are able to reduce the volume of the normal tissue that is irradiated, and also we have the possibility to spare lymphocytes inside the body of the patients, and so to avoid the lymphopenia, that is one of the crucial points of immunosuppression of the conventional radiotherapy. So I really think that this would be a very good direction for the future clinical trial and for the future translational studies regarding heavy ion radiotherapy, and to also thanks to the combination of this treatment, for example, with new drugs. And this is my last slide on the idea that I want to leave you on particle radiation biology. We know really few things about radiation biology in general. I'm not talking only about carbon ions or high latent radiation, but if we consider carbon ions or high latent radiation, we really have a poor knowledge of the properties and of all what is happening inside the cell and tissue and the body that is exposed to this kind of radiation. And these are all the topics that I guess I really recommend for future studies and on radiation biology for particle therapies. And I like to quote a statement of this paper where actually Marco Durante and Walter Tinganelli say that radiobiology of densely ionizing radiation is so markedly different than photons that charged particles should be regarded in radiotherapy in much the same way as a different drug is treated in medical oncology. So it's true that we are talking about radiation, but we are talking about a totally different type of radiation that activate different cellular mechanisms. So again, I will start from the beginning, say that I will go back to the beginning, say that it's not only a matter of double strand breaks and the DNA damage, but different activation of pathways inside the cells. And my recommendation for future works, some came already yesterday during the discussion with Eleanor Blakely, is that we need to move to more in vivo-like condition. Cells are not flat in our bodies, as I said yesterday, Eleanor. We have to mimic as much as possible the tumor microenvironment. So 3D models are really welcome now, and we have plenty possibility to choose different 3D models like scaffolds, synthetic or biological scaffolds like organoids and something like that. And also consider the interaction between different cells because tumor is not made only by tumor cells. So we have to consider also endothelial cells, fibroblasts, and all the cells that populate a tumor and that are irradiated as well in the treatment plan. And then I also suggest, because it's not easy to use in vivo models, that we should take advantage of what has been already collected by, let's say, classic biology, and to move towards alternative in vivo models, like, for example, zebrafish or Inovo models that may help in avoid some ethical restriction that are quite complicated to deal with, but they can give you very important information for concerning in vivo. And then my final recommendation is talk, speak, and discuss with your colleagues from all the specialties and professionals, because it's really important to interact, to exchange ideas, because radiobiology is not only biology and it's not only physics. It's also clinic, it's also chemical, and even inside biology, there are different points of view that has to be matched together to give very broad overview of all the effects of this relation. And thanks a lot for your attention. Nicolas?
Thank you very much, Angelica. Thank you.
Okay.
A very interesting talk. All right. So we have the opportunity to ask Angelica a number of questions. Are there any questions? Please indicate through the reactions and raise your hand through Zoom, or you can use the other tools that you have at your disposal. Okay, I can see that Celine, Carly. Is that the way I pronounce your name, Carly?
Hello, yes.
I hear the flow. I can't hear you clearly though. Is there something wrong with your mic, maybe?
Oh, it's better.
Not really.
You're metallic.
Okay, it's a bit. Yeah, until you fix it, I'll ask the second person. I'll come back to you. Okay, so Kyung Dong Choi, can I give you the floor?
Sure, thank you. Angelica, good to see you again, and it was a very nice presentation. So, do you hear me?
Yes.
Yes, yes.
Okay. So my question is that, okay, you know that I'm a physicist, and what I really want to know is that the biology seems to be very complex to me. So I want to know what is the essential experimental setup for doing the biophysical research in the treatment facilities.
Thanks a lot, and nice to see you here. And I think that the starting point is that physicists has to talk with biologists. That is happening, that not always happening. And so the first point is that we have to discuss on the models parameters. Because as I told you, and as I said during the lecture, for example, cells are not flat and cells do not have all the same shapes, for example. So already starting from the very basic things that we have to upgrade the models or to consider into the models the fact that we are talking about totally different systems. And concerning the ideal experiments, I would say that we still have to keep in vitro experiment because it's the easiest way to keep the experimental condition as much controlled as possible. And that is what you need for your mathematical models. But I also think that on the other side, the mathematical models and the physicists can be of very great help, for example, in moving towards 3D models. Because what I'm facing now using 3D models is that we as biologists have really, let's say, not such a big background on how to deal with this 3D structure. So I really encourage to interact biologists and physicists in planning 3D experiments together. Also with co-culture, but I think that will help so a lot.
Thank you very much. I was writing down, so I reacted a little bit. Thank you very much.
Okay, you're welcome. Ciao.
Ciao.
Thank you, Angelica. Okay, so we pass on to the next question. Celine, are you ready?
Can you hear me now?
Yes.
Yes.
Okay. I'm super sorry. Thank you again for this amazing talk. I have a very specific question. You mentioned the mTOR signaling pathway right in the beginning, and I remember that this also negatively influences the autophagy pathway. And so my question is, do carbon ions have a different effect on the autophagy pathway? And is there research on how this influences the whole immune response of the cell?
Yeah, there are, as I told you, let's say that molecular biology has started not so many years to be considered in radiation biology. So we are starting to collect data, and actually the first data that we have is that, yes, there are different activation concerning carbon ions, and in particular highlight radiation. We still don't know how it can be reflected on the immune response or in the immune properties of this radiation. But you know, molecular biology is a very huge field, even without radiation. If we consider that we have to match molecular biology plus radiation plus different types of radiation, we are really facing to a new world, and we are not ready at this moment to have a conclusion, but definitely is a field that we have to investigate.
Okay, thank you very much.
You're welcome.
Thank you, Angelica. So next was Matthias with.
Yes, hello. I have a question regarding the cancer stem cells. Is it possible to identify them? And if so, how do you identify them? And is it possible to identify them in vivo?
Yeah, okay. Unfortunately, there is no one unique marker for all cancer stem cells. There are CDs markers that are receptors that I don't know your background, but actually there are some receptors on the cell surface that can identify these cancer cells. So for example, there is CD133 that is a quite widespread marker for different type of cancer stem cells. So actually there are antibodies able to identify them. But as I told you, it is in principle possible to identify them in histologically speaking, because we have antibodies to detect them, and in principle should be also in vivo. But again, we don't have in our hands a marker that is unique for all the tumors, for all the cancer stem cells, and also for all the patients, because again, we have the problem that every patient is different from the other one. So maybe a marker that is suitable for one is not suitable for the other one. But definitely the imaging of these cancer stem cells can help in tailor the treatment also, for example, with different type of radiation of different ions than carbons.
Thank you.
Thank you.
Sorry, but it is we don't know exactly one single that can suit for all of them.
Thao Nguyen Pham.
Can you hear me?
Yes.
Yes.
I have a question about regarding the immunogenic effect of radiation, and in particular the lymphopenia. What do you think about the mechanism underlying radiation-induced lymphopenia? Because as I know, lymphopenia is occur even if in the brain irradiation, when there is very few bone marrow or very few lymph node or secondary lymphoid system. So as I know, that maybe is the effect of radiation on the circulating blood, but I see that the amount of blood in the radiation field is very small, but radiation-induced lymphopenia could occur in a very long term after brain irradiation. So what do you think about this point?
Yeah, it's a really good point, because what we know about the lymphopenia is derived from the analysis of the blood that have been collected from the patients that have been treated. So we have evidence on that, and we have speculation on the fact that regarding to the physical doses, to the physical properties of high-level radiation, we are going to give very low doses to normal tissue and blah, 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 what I said. But you have raised a very good point, because there should be also some other biological mechanism behind, because when we treat a very small volume or tumor location with very low amount of lymphocytes and blood flow, there should be something else that is occurring, and we don't know that. It could be something, definitely some cytokines or some, let's say, humoral factors that are released after irradiation with high-level radiation. But we have the evidences that are made by analysis, but at the moment we don't know the mechanisms that are under that. Sorry.
Thank you very much.
Thank you, Angelica.
Very good point.
Okay, we'll move on to the questions that Chris Sapps has.
Thank you for the very nice presentation. It's sort of two interrelated questions. It's more about translating it all to the clinic. So firstly, the OER, I've heard that it's not that straightforward for the modeling as it is with the RB, and also that the dose dependency of the OER is a bit harder than RB. So sort of question on that, and OER translation into clinic. So do we do just some assumptions of some specific like regions in the body, or we use like PET hypoxia imaging for that to sort of include those points?
Yeah, you know that this question should need a lot of time to be addressed. But anyhow, you know that hypoxia is one of the main topics that is under study for the new, let's say, the new ions, because we know that inside the tumor we have hypoxia and not hypoxia region, and we can have an idea on the region. So the idea is to have a mixed treatment with different radiations in order to overcome the hypoxic radio resistance. So for example, the use of oxygen ions that is supposed to deal better with this hypoxic region, because as I told you and as I saw you, carbon ions is able to reduce the oxygen effect, but is not able to avoid it at all. So if we consider then cancer stem cells are likely to stay in that hypoxic region, we have definitely we definitely need to stress the doses and the biological effects on that hypoxic region. And how to deal with that from let's say physical point of view, I can say just trying to exploit as better as possible also the radiosensitizer effect, for example, radiosensitizer drugs, for example, because I haven't touched this topic, but another huge word is the interaction of high-density radiation with the drugs and with other radiation as well, because at the moment we don't know the interaction of this new drug in radiotherapy that is high-density radiation with new with the radiosensitizer that is used in in conventional radiotherapy. So it's I think that the direction is to use different ions to overcome this hypoxic resistance. For the moment, I mean, we are in very good direction, and we can continue in that direction.
Thank you, Angelica, so much. I'm afraid we will have to stop here because we have our next speaker that is scheduled at 10 o'clock.